All right, welcome back everyone. So we're going to now look at these kind of three main stages in a lymphocyte's kind of life cycle, right? And we'll take them one at a time and we'll expand on various ones. So we're going to first look at the formation of lymphocytes. We're then going to talk about how and where they are activated, and then what is their effector response, right? So what do the T cells and B cells, what do they actually do for our immune system, right? So if we look at our formation first, right? So the formation of our lymphocytes, we talked about this a little bit in our lymphoid lecture, right? So when we did our lymphatic system, we've discussed a little bit of where our immune system cells are formed, right? So all the blood cells, they're all formed in the bone marrow, right? This is where they're formed. Where do they go to become activated ultimately? Um, um, or I'm sorry, where they go to not activated, where do they go to mature? The B cells are staying in the bone marrow, right? And the T cells are going to the thymus, right? And so those are the bone marrow and the red bone marrow and the thymus. Those are our primary lymphoid structures, right? So red marrow and our thymus, and then our secondary lymphoid structures. Those are all of our other ones. So you think like spleen, lymphatic nodules, lymph nodes, things like that, right? Mucosa-associated lymphatic tissue. All of those are the secondary structures where the cells ultimately go to become activated, right? And so here you can see the activation, right? And we'll talk in more detail about kind of each of these various processes. But they can go to our lymph nodes, spleen, tonsils, mucosa-associated lymphatic tissue, the MALT, and they're going to ultimately become activated, right? So whether that's T cells or our B cells, which are going to differentiate into something called a plasma cell, and then the plasma cell secretes antibodies, right? And so this is where they're exposed to antigens and they ultimately become activated. They're going to replicate, they're going to begin dividing, right? And we're going to talk about that process called clonal selection in a little while. But this is where they're ultimately going to become activated, right? So just some questions to consider. So if we look at our T cells in particular, right? So where do we make our T cells? We already said that they're made in the bone marrow, right? They originate in the red bone marrow. They go to the thymus to ultimately mature. And here they have those CD4 and CD8 proteins, right? So remember, CD4 goes with which MHC molecule? It goes with MHC2, and CD8 goes with MHC1, right? So remember, when you multiply the 4 and the 2, you get 8. When you multiply the 8 and the 1, you also get 8, right? So how do you get the T cells, right? How do you form the T cells and mature them to the point that they are going to recognize self antigens as being self and ignore them? And how do you get them to where they're going to recognize foreign antigens as being foreign and ultimately initiate an immune response, right? So these processes are called positive and negative selection, right? Positive and negative selection, right? So we'll first talk about positive selection and then we'll do negative selection, right? So positive selection, this implies that you are selecting cells that do something, right? So positive, they're actually doing something, and by doing that, you decide to keep them, right? So positive selection, these T cells, and these are essentially think of these as like immature T cells, right? They're not fully formed yet. They're not mature. They haven't been exposed to antigens yet. They're just in the process of being formed and developed, right? So these are T cells that are, or thymocytes. These are just like kind of immature T cells. This positive selection in the cortex of the thymus, this is involving T cells being presented an MHC molecule and determining whether or not that T cell will bind to and recognize the MHC molecule, right? So because this is positive selection, cells that do bind to the MHC molecule are kept, right? So you have a thymic epithelial cell. It shows an MHC molecule to a T cell. If that T cell binds to the MHC molecule, that's good. It keeps the keeps the T cell. If it doesn't recognize it, right? So if that T cell receptor does not bind to the MHC molecule, then this implies that that T cell would not be able to bind to the molecule and antigen in circulation in the event that it was presenting a foreign antigen, and it wouldn't initiate an immune response, right? And so you need cells that are going to be able to do that, right? So in this case, if it does bind the MHC molecule, it survives. If it doesn't, you kill it, right? So it's destroyed via that process of apoptosis, right? So in this case, positive selection, you're keeping cells that do bind, right? You're keeping cells that do bind, right? This is in the cortex of the thymus. Negative selection is exactly the same except that it's totally opposite, right? So in this case, this is in the medulla of the thymus, still in the thymus. Now we're keeping the cells that don't bind or don't recognize, okay? So in this case, we already understand that if the T cell receptor binds to the MHC molecule, it's kept. That's good. That means it has the ability to at least bind to the cell, bind to the infected cell, the antigen presenting cell, whichever cell it happens to be. Now, those same T cells are now presented a self-antigen, right? So a cell in the thymus is presenting these kind of immature T cells. They're presenting them with a self-antigen, and they're seeing if whether or not the T cell is going to recognize and initiate an immune response against the self-antigen, right? So in this case, if it recognizes the self-antigen, that's bad, right? This can imply that the cell could potentially recognize a self-antigen as being a foreign antigen in the body and initiate an immune response against it, right? And so that's not what we want, right? If we want normally functioning immune cells, right? So now we are selecting the cells that don't bind. So does it recognize the self-antigen? No, it doesn't recognize it as foreign. That's good, therefore you keep it. Right. So the first one, you're keeping the cells that do bind, that do recognize. Second one, with negative selection, you're keeping the cells that don't. Right. And again, if it did, it would likely cause some type of autoimmune disorder, right, or some type of autoimmune reaction, right. And so this is something called central tolerance, right? We are creating cells that are ultimately going to be able to tolerate our self-antigens and then initiate immune reactions against foreign antigens. Okay. Now, then comes the question of whether or not the cell becomes cytotoxic or helper T cell, right? As we've mentioned, right? So, in this case, throughout this process of positive and negative selection, the cells are sometimes termed a double positive, right? Meaning they have both CD4 and CD8 proteins. Okay. In the thymus, the cells will then lose one of those proteins. It'll either lose the CD4 or it'll lose the CD8. If it loses the CD4 and it keeps the CD8, it's a cytotoxic T cell. If it loses the CD8 and it keeps the CD4, then it is a helper T cell, right? And so then you can then say, okay, which of those cells are going to bind with the various MHC molecules, right? You can say, okay, CD4 is going to bind with MHC2 on antigen-presenting cells, and CD8 is going to bind with MHC1 on all nucleated cells, right? And so this is this process of losing one of these CD4 markers, right? So at this point, our T cells are leaving the thymus. They're going to those secondary structures. So again, think like maybe spleen, lymph node, lymph nodules, tonsils, things like that. And they are immunocompetent. This means they have the ability to bind an antigen, but they haven't yet, right? So they're immunocompetent, but they're naive, right? So meaning they have the ability, they have the qualifications, they have the right, hardware, but they have not yet actually encountered an antigen, right? So they're called a naive T cell, right? There are also cells called regulatory T cells, and these are CD4 cells formed from T cells that bind self-antigens, and these are going to inhibit immune responses to some degree in something called peripheral tolerance, right? So function and tolerance outside of the primary lymphoid structures, this is called peripheral tolerance, a form of self-tolerance. So again, helping to prevent those autoimmune type of responses, okay? So let's stop here with this short section, and then we'll pick up in our next one talking about activating those T cells and causing them to divide, and then ultimately we'll get into what their effect is.